Great pleasure to introduce Professor Philipp Kukura from the University of Oxford. Professor Kukura studied chemistry in Oxford until 2002 and did a PhD from the at UC Berkeley in Rich Matthews on femtosecond simulated Raman and other ultrafast spectroscopy techniques before moving to ETH Zurich to study nano-optics. He is a pioneer in a variety of technologies, femtosecond simulated Raman being just one of them, but things like mainly light scattering techniques, of which we have a great deal of use in biology and is going to be really key to, I think, the next 20 years of single molecule unlabeled imaging is kind of starting right now. So without further ado, I'll just kind of hand over. So just to be clear, if you want to ask questions in the chat, Professor Kukura has kindly said he will keep an eye on the chat and ask it, and answer them at suitable times during the presentation, or you can wait until the end. And if you would like to ask your questions in person, we can invite you up and you can unmute and turn your camera on if you would prefer that. But without further ado, I'll hand over.
Thank you, Chris. Let me do the screen sharing. Right. Okay, so thank you for having me. Would have loved to be there in person, but I think we're not quite there yet. So getting closer and closer. I did manage to give a talk in person the other day for the first time in 1.5 years, which was quite an experience. One tends to forget. But for now, we'll stick to the digital world. As Chris has mentioned, I will talk to you about light scattering. And specifically about a technology that we've developed and been working on, for quite some time. And the principle behind it is relatively simple. So let's say that hypothetically you could hold up one protein, you could shine light on it. There will be some form of interaction between the light that hits the protein. And as a result of it, for example, you could envision that there would be a shadow, right? That would be one thing that could happen, or the protein will scatter some light. It's really up to you. That interaction is going to be weak, but nevertheless, it's not going to be zero. And whatever interaction you may have, depending on the nature of it, you may agree with me that if you have two proteins, you'll have twice the interaction, right? One protein weak, two proteins twice as weak, if you like. And if you accept all of that, then you may come to the conclusion that if you could actually measure this interaction, quantify it, detect it, and you could do that with high precision, then you could tell the difference between one and two proteins. And because two proteins weigh twice as much as one protein, maybe you could not only image and detect individual biomolecules, but you could also measure their mass. And essentially, that's the idea behind what we call mass spectrometry. Right, so the next question you may ask, brilliant thought experiment, well, who cares? Why is this useful? So here's a slide that is kind of the epitome of what used to drive me crazy about biology when I was in high school and was an undergraduate, which is always this diagram with lots of blobs and acronyms and arrows, and I couldn't really figure out what's going on. But ultimately, this is how we understand a lot of processes in biology. And I'm a chemist, so I look at this and think, well, actually, it's not so different from what organic chemists think about when they draw reaction mechanisms, except there, what they worry about is where do electrons move and where do you make and break bonds. And in biology, it's not so much not necessarily making or breaking covalent bonds, but what you're dealing with is some kind of choreography, some kind of sequence of interactions between biomolecules. So this example here is proteasomal degradation. And the idea is, here you have a happy folded protein. If your happy folded protein becomes unfolded for some reason, stress or heat or something like that, this is an issue because these kind of things can aggregate. And as you will know, a lot of neurodegenerative diseases are associated with protein aggregates. So the body or nature needs to have a mechanism by which one gets rid of this. these kind of proteins. And the way it does that is there's a whole series of enzymes, and essentially they target misfolded or unfolded proteins. And when they bind, they essentially attach a tag, ubiquitin, and not only do they do it once, they actually attach a whole code. And once there's a right code and an unfolded protein, it gets recognized by this macromolecular machine, this proteasome, and it eats up the misfolded protein. At the end of it, it's gone. Right, it's a big success. Now, I've drawn this whole mechanism. You can, how do you get there? Well, in a very abstract way, you could say, there's a few questions I need to be able to answer in order to arrive at a mechanism like that. So you need to know what you have. You need to be able to tell the difference between E1, E2, E3, the proteasome, your substrate. You need to know how much you have, right? I started with some unfolded protein. I ended up with none. How does it change as a function of time, and what interacts with what, right? So here you have an E2, E3 complex, for example. Here you have this whole machine. And in many ways, a lot of the technologies that are out there that we use in order to understand these kinds of mechanisms fundamentally answer or help us answer exactly these kind of questions, right? What do I have? How much? How does it change? And what interacts with what? And the reason why this is important is not just because we, out of scientific curiosity, we want to know how, misfolded proteins get degraded, but it's also important because, when something goes wrong with this process, that can lead to disease. So we want to be able to understand the process so we know how it changes when it's gone off. But also, for instance, now one of the big future avenues in pharma is to develop molecules where you can small molecule glues that essentially pick specific proteins and attach them to these ubiquitination machines. And if you think about it, what you've done there is you've developed an approach by which you can specifically target proteins for degradation, even though they're not unfolded, right? So this is it's a new class of drug that people are working on, and ultimately depends on understanding the underlying mechanisms. All right, so how do you answer these questions? Well, you need some machines, ideally, that give you answers to these questions. And there's lots and lots out there, and I'm not going to go through all of them. I'm just going to pick the ones that suit my narrative, if you forgive me. So one is mass spec, right? And mass spec is awesome because mass spec measures mass very, very accurately. And protein A has a different mass than protein B, has a different mass than protein C. So even in a mixture, in principle, you can tell different biomolecules apart. You can find out how much you have, and maybe you can even find out how they interact. And if you wanted to complain about it, you might say, well, it's an ensemble technique, so I always look at lots of stuff, and it's also in the gas phase, which maybe could have some issues when you're looking at proteins interact that normally do that in solution. Your second option is to use fluorescence. And what you do is you attach a label. And the amazing thing about that is now you can really watch individual molecules, right? So you can see one molecule attached to another and then fall apart. And you can do it in a dynamic fashion. You can do it in water or in buffer. But the one thing that maybe is not so nice is that most molecules are not fluorescent by nature. So what you have to do is you have to label them, right? So it's not really a universal approach. And then finally, scattering. Scattering is great. Everything scatters light. And of course, you can get atomic structure information, whether you're using electrons or whether you're using X-rays. Now, you could get greedy and say, right, I want all of that. I'd like to have the specificity I get from mass. I'd like to use light because it's dynamic, and in principle, single molecule, and scattering because it makes it universal. So could you do mass-sensitive light scattering, which is essentially what I described to you at the beginning of the talk? And the question you might ask, okay, if this is so attractive, why hasn't anyone done it? Or why is it not much more widespread? And the simple reason is that proteins are very small. So they're very, very small compared to the wavelength of light, which means the interaction between the protein and the light is going to be very weak and essentially impossible to detect, or at least that's what we all thought 5 years ago. Now, is that really true, right? Because if you think about it, protein is 5 nanometers in diameter, and your diffraction limit is something like 200 nanometers. So you have a factor of 40 difference between the size of your diffraction limit and your protein. So yes, the interaction is going to be weak, but is it necessarily zero? And the answer is probably not. Now, you've probably all seen this at some point. And of course, what you see here is an oil film on top of water, and you'll see slightly different colors. And the reason why you see slightly different colors is because of interference. And you can do what's happening is you've got light from the sun, that's white light from the sun, that hits the interface, and there will be some reflection at the air-oil interface that will hit your eye. And there will be another reflection at the oil-water interface, and that will hit your eye. And the two reflections will interfere with each other. And depending on the thickness of this oil film, that means that some wavelengths will be preferentially transmitted and some wavelengths will be preferentially reflected. And what that is, what wavelengths are preferentially reflected, will depend on the thickness of the film. So if it's 100 nanometers, then you'll see mostly yellow light if you start with white. But if it's 95 nanometers, it'll be green. So this is awesome, right? I mean, you're using the sun as a light source, and you're using your eyes as a detector, and you can clearly and easily see the difference that is of the order of the size of a protein. It's 5 nanometers. So why should this be so hard? Well, where I cheated, of course, is that when you look at that photo, if you look at that oil film, you're integrating over large areas. So yes, it's 5 nanometers in one dimension, but it's many, 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 many centimeters laterally. All right, fine. So why don't we just add a lens, right? If I add a lens, if I add a really good lens, then I can get close to the diffraction limit. So maybe I can focus light down to 200 nanometers. So I can maybe do exactly the same experiment. And if I replace the sun with a really good light source and my eyes with a really good detector, maybe I can do better. All right, so here's that experiment. So your sun has become the laser. Your eyes have become a digital camera. Your lens is a microscope objective that focuses really tightly. And the only difference is now you shine light onto a glass water interface, and you look at the light that comes back rather than shining light from the top and looking at the reflection. But you can see why those things are essentially identical. And what will happen, light will hit this interface. Some of that light will get reflected because you have a change in refractive index here. And then your protein will scatter some light, and both of those will eventually hit your detector. All right, so what happens when you build this magnificent piece of equipment is what you see here on the right. So this is what you will see on the camera. And this might explain to you why nobody's bothered doing this very much, because you do all of that, and then what you've done is you've built a really expensive beam profiler. Because essentially what you've done is you've taken an image of your laser beam. And you can see that, just over this field of view, the intensity varies by about 90% from left to the center all the way to the right. So how on earth are you going to see the tiny, 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 tiny little scattering signal that you're going to get from a single protein? And the answer is forget it. It's never going to work. All right, but you can do one thing, which is if you have a good laser, right, then this isn't going to change. Right? It's completely constant because that's just the profile of your laser. So if I put my interface, my sample into focus, or this glass water interface into focus, and if I translate it, then I can actually measure this illumination profile. And when I take an image, I can just divide each image by this illumination profile. And when you do that, you get this thing on the right. Okay, so just take a commercial cover slip, put water in it, subtract the background, if you like, that is given by the laser beam, and you get this here. And you say, oh, well, that looks horrible. And you're right. So this is essentially a speckle pattern. But now you see that essentially what you have is on the scale bars are about a micron here, right? So on the 200 nanometer scale, you have changes in reflectivity or variations in reflectivity on the order of a few %, right? And the next question is, so what does that mean, right? What is it? All I know is when I translate my cover slip, this moves. All right, so it probably has something to do with the Coverslip interface, and in fact, that's the case. But how big is that signal? Well, it turns out that signal is about the same size as what you get from an actin filament. So an actin filament is essentially a protein chain, right? So you have a 40 kilodalton protein, you line it up, a protein chain, and this protein chain you can just about see on top of the speckly background here, right? So this is literally just put it on glass, it adheres to glass, you get these little lights. All right. Now you can do a calculation and you'll come to the conclusion that your detection limit is something like 70 actin molecules, all right? But your detection is entirely limited by the fact that you have this annoying speckly background that you get from the cover glass. But I can measure the annoying speckly background really well, right? So in principle, my detection sensitivity is better than what is limited by this background. It's just, you know, can I do the same subtraction when I went from here to here? Can I now subtract the speckly background as well? And maybe I can see a signal that is smaller than 70 actin molecules. And that's essentially what we did. So you have actin here, and then there's a protein called myosin 5, which is a molecular motor, walks along actin if you add ATP. So if you think about it in terms of subtraction, you have this image here. You add myosin, myosin will bind to the actin, and then in the presence of ATP, it will walk along the actin. So when I do an image subtraction, I can subtract the static image and the static actin, and all I should see is myosin arriving, moving, and departing. And here's the movie, right? So you can see where the actin is, and you can see that where the actin is, there's black dots and white dots, and black dots means that proteins have bound, and you can see them move, all right? And for each of these dots, we can quantify the signal and then make a histogram of the signal, and you know it's something like 0.1%, fine. And sometimes we can see this, right? You can see how the myosin is jerking forward. And turns out the step size that we see is 37 nanometers, and that's the step size of the protein. So what we're seeing is probably a single protein. The other thing we can do is we can look at this signal here, and it turns out to be a fraction of the signal from the actin filament. And in fact, if you divide the mass of the actin in a diffraction limited spot by the contrast and multiply it with the mass of myosin, this contrast here corresponds to 550 kilodalton, which is the mass of one myosin molecule. So what does that show you? Essentially, you shine light on a cover slip, and you look at the light that comes back, and if you do it really sensitively, you can see the change in reflectivity effectively of the interface as a consequence of the presence of one protein. And the size of the change in reflectivity is proportional to the mass, all right? Okay, so what's the problem? The problem is this, right? So yes, I told you there's little black dots and they move around, and there's some white dots, and you will look at it and say, oh man, but you know, okay, fine, you see some black dots, but what are you going to do with that? And in fact, that was the response of the referees when we originally tried to publish this paper. And you know, in hindsight, unemotionally, probably they had a point. So why does this look so bad? Well, the reason why it looks so bad is because the signal is really small, right? So it's a fraction of a percent. And essentially, it's really hard to acquire enough photons so that you can lower the shot noise or the background noise sufficiently so that you can see these little black dots. And the other problem is that you have all of this background. So you have lots and lots of light that comes from the interface between glass and water, which is the light that is being reflected, and very, very little light that is being scattered. So your detector, you know, will see a purely reflected term, purely scattered term, and interference term, and this purely reflected term is enormous compared to the signal, which is this interference term. So is there a way to reduce this? Because if you did, then what you would do is you would increase the contrast. And the answer is yes. So we worked on a whole bunch of different approaches where we try to see really small signals in the microscope. And essentially what happens is if you have a point light source at the interface between a high refractive index and low refractive index, in this case glass and water, it turns out that most of the light that is scattered by this object actually goes to the high angles. So it isn't isotropic, the scattering. Most of the light, like 90% of the light, goes into these two wings. So you have this kind of pattern. All right, so if that is the case, what if you build a setup like this? So you take a laser, you shine light onto your sample, and rather than just using a beam splitter or something, you use a piece of glass with a little bit of silver on it. That will reflect the laser beam, and you focus your laser so it only goes through the center of the objective. That's a standard way of creating wide field illumination. The light will get reflected, will come back, and it will get reflected again by the silver mirror. And if you get the thickness right, then only a very small fraction of the light that's coming back will get through this little mask. But because your scattering is all on the outside, all of the scattering will make it past this mask. And essentially what you're doing there is you're changing exactly this ratio between the reflected term and the interference term. And when you do that, now your single molecule images look like this, right? So suddenly your contrast is much bigger. You can see that something like a factor of 10 bigger in this particular case. And because your contrast is bigger, you can use more photons illuminating your sample. You can reduce the shot noise, and suddenly you get really pretty images. And here's how the experiment actually works, right? So all you do is you have a microscope cover slip. You have some buffer. Here's your buffer, right? You can see the objective here. You take a few microliters of your solution. You add it with steady hands to your buffer. There you go. And then your molecules will diffuse, and some of them will non-specifically bind to the surface. And here are your molecules. Okay? And every single one of these little black dots is one protein, or in this particular case, one oligomeric complex of the protein. And this is exactly how the experiment actually works. So where does the mass come into this? Well, the light that we detect, or the magnitude of the signal that we detect, is proportional to the polarizability. The polarizability is proportional to volume and the optical properties of your sample. And of course, your volume is proportional to mass. Now, all proteins are made of the same amino acids, so the optical properties are going to change very much. And in fact, if you look through the what's called the specific volume, the density of proteins all pretty much the same, right? 50 kilodalton protein, 4 nanometers, not like some 40 and some 0.4. So if the density is constant and the optical properties are constant, that means that your polarizability is proportional to mass. All right, so is that true? Well, here's a protein that is highly monodispersed. So this is a 20-former, so 24 proteins that make a nice little symmetric ball. And it only exists in the shape. And when you add the sample, you get, again, a black dot for every protein that lands on the surface. And you can see from this movie that every single one of those signals has the same intensity, right? One thing to mention, the black dots appear and they disappear. That's not because proteins arrive and leave. It's because we have to do this background subtraction, and we do this on the run. So we only see proteins when they actually arrive. Before they are not there, and afterwards they become part of the background. You take a slightly different heat shock protein, HSP27, that's polydispersed now. So you get anything between 54 and 1,000 kilodalton. You run the movie, and now you see very different behavior, right? So some dots are really, really intense, and some dots are so faint that you almost can't see it, which tells you that there seems to be a relationship between the size of the signal and the mass, exactly as I predicted. All right. Let's take a slightly more challenging system, but very boring biologically. So you take BSA. BSA is a 66 kilodalton protein. Here's a movie of BSA. And again, you see some really faint dots and you see some really dark dots. For each of these dots, we fit it and we determine the size of the signal. And depending on how big the signal is, that determines where the dot is on this axis. And depending on when the molecule landed, that's when the dot is on this axis. And when you do this for this movie, you see all of these dots, and you can clearly see there's a band here, and there's another band here, and then when you integrate up into a histogram, here's another band, and maybe there's a fourth one. And then you look at the contrast, and you see, well, this is about 0.04, 004, 0.008, 0.012, 0.016. They're all equally spaced. So when would you expect to see equal spacing? Exactly in the same way as I started, right? One protein produces a certain kind of signal, 2 proteins twice the signal, 3 proteins 3 times, 4 proteins 4 times. So indeed, this is the monomer of BSA, this is the dimer of BSA, this is the trimer of BSA, this is the tetramer of BSA. So the scaling seems to hold. And in fact, you do a whole bunch of different proteins, lots and lots and lots and lots of proteins. Here's a sequence mass, and you can measured mass, and you can see that the correlation is really quite excellent, about 2% mass accuracy. Now, of course, that's not as good as mass spec is, but actually for most things it's plenty, because what this graph shows you is if you have a 100 kilodalton protein, on average you'll measure between 98 and 102. That's plenty to give you confidence that you're looking at the right protein. And it's really this combination of the resolution, being able to tell the difference between 1 and 2 and 3 and 4, or 2 bits that are not bound to each other, 2 bits that are bound to each other, and mass accuracy, so really being able to measure the mass accurately. That's what we then call mass photometry. All right, so we published that about 3 years ago, and since then we've spent quite a lot of time trying to figure out whether and to which degree this is actually useful, right? So there we worked on polypeptides. So what can you actually do with this? And here's some examples. So one would be assembly, right? How do you go from individual proteins to, let's say, an assembled oligomeric structure? And this may be important, for example, if you're interested in viral vectors, right? Some of your vaccines are based on adenoviruses. So you have to assemble these vectors. You have to make sure that they're assembled properly, et cetera, et cetera. So here's one thing, one application. Then membrane proteins. Membrane proteins are really important as a drug target, but horrendously annoying to study because, of course, they want to be in membranes and not in solution. So you always have to solubilize them. And then things get heterogeneous and tricky. Well, it turns out this technology also works just fine with membrane proteins. And then finally, nucleic acids, clearly very, very important, both by themselves in terms of their interactions with other proteins or with proteins. And it wasn't clear to me at the time what kind of signal you would get, because if you have a globular protein, you might imagine that there's going to be some scattering because it's kind of like a nanoparticle. But now you have a molecular wire. What's the refractive index of a molecular wire? Wasn't quite clear to me what we would actually get. And it turns out that you get a slightly less signal per mass for nucleic acids than for proteins, but it's not a big change. So you can see still this very nice linear relationship here between the number of base pairs and the signal that we measure. All right, so I'm going to just talk about a few examples here. I'm just trying to check where I am with time. One is to look at interactions and kinetics. This is clearly important, for lots and lots and lots and lots of applications. And the most simple one might be, if you're looking at antibodies and how they interact with targets. And if you think about how you define an equilibrium constant, right? What you need to know is how many free species you have and how much complex do you have. And if you can measure these three things, then you get an equilibrium constant. Well, this has mass A plus B, this has mass A, and this is mass B. If I can see and count all of these things, then maybe I have a really simple way of looking at affinities. Structure, right? We always draw, draw a proteasome and says, oh, this is what the proteasome looks like. But this is a massive machine. Is this really what I get? And if you think about it, for a lot of structural biology, yes, your cryo-electron microscope will give you a structure of pretty much anything if you can freeze it, but that will only work if most of your particles actually are assembled. So a big challenge is to make sure that they are assembled. And of course, if you can measure the mass of your particles, if they're all at one mass and a target mass, then you know that your sample is in good shape. And then finally, everything I've shown you so far requires proteins binding non-specifically most of the time to a glass surface. Can we do the same kind of thing on bilayer membranes? All right, so here's your interaction. I quite like this little diagram that Fabian Soltemann made at the time, or had made at the time. And it really works like this, right? You count, how many do I have of this? How many do I have of that? And I should be able to get the KD. All right, so here's a super simple example, tubulin, which makes microtubules. It's a dimer, as far as everyone is concerned, but it's a non-covalent dimer. So if you go to low enough concentration, everything will eventually fall apart. So if you take tubulin, you measure the mass distribution at 20 nanomolar, you see the spectrum here. So there's two peaks. First one is the monomer, and the second one is the dimer. So you can count how many are in the dimeric form and how many are in the monomeric form, and from that you can extract the KD. So in this case, 8.4 nanomolar. And now you say, I don't believe you. How could you possibly do this in one measurement? It can't possibly be true. So you say, okay, fine. So what I'm going to do is I'm going to do this measurement at lots of different total concentrations of tubulin. If I'm below the KD, I would expect everything to be monomeric. And if I'm well above the KD, I would expect to mostly have dimers. And sure enough, that's what you see. So at 1 nanomolar, almost everything monomeric, and you see the dimer peak grow and grow and grow. And for each of these spectra, you can measure the KD, and these are the measurements. And the answer you get, 8.5 nanomolar. And if you still don't believe that, you can kind of do a more traditional titration curve and fit the whole thing. And again, you get pretty much the same answer. So in other words, this concept of, oh, I just look at species and I count them and I get the KD, at least for tight affinities, works well. The other thing you can do is you can get kinetics, right? So option number one is you mix something together at high concentration, you dilute down to low concentration, you watch things fall apart, or you mix things together at low concentration, and then it's going to take time because of diffusion for complexes to form. And essentially, with that approach, you can get both on rates and off rates. And if you get on rates and off rates, you can calculate the KD. So here we calculate on rates and off rates, and we get a KD for 30 picomolar. If we actually measure it, we measure 50 picomolar. Here we measure on and off rates, we get 1 nanomolar, and again we measure it, and what we measure is about 1 nanomolar. So you can either use the kinetics or you can use the distribution between the two peaks. All right, so let's talk about structure a little bit, right? So here's, this APC complex consists of all of these different proteins, and you want to get the structure of it, so you need to actually make sure that that's what you have. And as powerful as cryo-EM is, if you have lots of different things, it's not going to be so simple to get a good structure. So there's a lot of interest in getting the right stuff in there. So we took, one particular system here. This is a giant transmembrane protein with both a transmembrane domain and a soluble domain. And we just looked at it and we saw a bunch of peaks, right? So on 3 main peaks, there's this one, which is soluble bit, this one, which is almost the entire complex, and here you have the entire complex. And you took the same sample and stuck it in an electron microscope, and the predominant species you see are exactly those. So you have the full complex, a slightly smaller one, and then only the soluble part. And the contrast roughly scales in the same way. Now, these are not the same particles, but the idea nevertheless holds. And you can then do things that lots of people have been, try to do. For instance, if you want to enrich a particular complex, so in this case, our collaborators wanted complex number 3, and one way to do that is to add chemical crosslinker. And sure enough, if you spend 5 minutes with glutaraldehyde, you see that this peak 3 comes up, and you also see that it shifts to higher mass. That makes sense. It comes up because you stabilize the complex, goes to higher mass because you're actually adding mass in the form of the crosslinker. So you can use this approach to relatively quickly enrich a particular species and check for it. And it also works for systems that are not structurally very, homogeneous. So this is cohesin. Lots of people are interested in cohesin, how it works. And one of the challenges is you can't really get a structure of it because it's all floppy. And this is what the EM looks like. But if you do the mass photometry, you can see that at least your sample was really quite pure. Most of it was assembled. All right, so we put this to the ultimate test, if you like, and we kind of went through an entire purification protocol. So you have the different purification steps, and ion exchange, size exclusion, etc. And usually what you do is you look at the gels, and then you do negative stain, and eventually try and figure out when you have something pure. So here is, as you get through the purification protocol, right? And you can see after the first purification, complex you're after is here at about 1.2 megadalton. So after the first purification, not so much assembled. Ion exchange, first fraction not so good, this fraction much better. Size exclusion, the first 2 fraction great, the next 2 fractions not so good. And here were 2 samples that our collaborators found in the freezer where they did lots of chemical cross-linking and purification, and sure enough, you can see slightly higher mass and really, really pure samples. And we took all of these samples, when you also stuck them onto a negative stain EM grids and did all the classification and counted the particles and then figured out the fraction of assembled particles. And the circle here is what we measured with mass photometry, and the triangle is what we measure with negative stain. You can see that they track really, 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 really nicely. And the main difference is this is super quick and easy and doesn't require an electron microscope. It takes you about 30 seconds. But you get the kind of key information that you're after. All right, so the final one is membranes. And you might ask, why do you want to do this on membranes or in membranes? Well, one argument might be that you have transmembrane proteins, GPCRs. There's bound to be interactions between transmembrane proteins. But if you solubilize both of them, what interaction are you actually going to measure? Because of course it has to break through this detergent shield. What you really want to do is ideally you would want to do this in a membrane. And the question is, how are you going to do that? Well, this is where Eric and Manish have come in. And they basically said, well, we measured because stuff binds and unbinds, but binding and unbinding is just a different version of a protein diffusing from here to there. And essentially that goes all the way back to the myosin experiments that I showed you before. So can we develop a better algorithm to extract this information? And essentially this is what the data looks like. So we took a membrane-associated protein called dynamin, made a bilayer, just threw the protein in, and then we saw these little black dots, and the black dots you can see move around. And just like before, you see some black dots, some really black dots, dark dots, and some really faint dots. So presumably you have the same kind of behavior going on as before. The difference now is that you can not only see that the dots are there, you can also measure how they move, and you can also quantify how long they stay on the membrane. So when you measure the distribution of contrast, you see this thing that you are now familiar with, right? A bunch of peaks that are all equally spaced. So if you plot the expected mass versus the contrast, again you get a nice straight line, which means you can now measure mass. There's high localization precision. You can track things for a very, very long time. If you can track things for a long time, you get long trajectories. From long trajectories, you can determine diffusion coefficients very accurately. But now you can do this as a function of mass, right? So here are your dimers, and they all diffuse quickly, and your tetramers that are more slowly, and your hexamers which are even more slowly, and your octamers even more slowly. And the trained eye, you will see, well, the relationship kind of looks like 1 over the mass. And indeed, you're right. If you plot the diffusion coefficient versus the 1 over the number of subunits, you get a straight line. And that makes sense, right? If you have a dimer, you have 2 fingers in the lipid pie, and it's diffusing relatively quickly. And then when you have a tetramer, you have 4 in the pie, so it's going to be slower because you've got 2 more, and 6 even slower, and 8 even slower. That's comforting on the one hand, but it also tells you that as the dynamin is assembling, the interaction with the membrane is the same every single time, because it affects the diffusion coefficient exactly in that kind of fashion. Similarly, you can plot the trajectory length, right? What kind of trajectory length distribution, you can fit that, and from that you get a rate constant for detachment. And again, that scales as 1 of the number of subunits. So indeed, if you have an octamer, lots of fingers in the pie, it will stay on the membrane for a long time. A dimer, very few fingers in the pie, only stays in the membrane for a very short amount of time. And sometimes you get to see stuff like this. So here you have, I think this is an octamer or something like that. It's diffusing around, and after some time, bloop, it falls apart. All right. So this is a dissociation event on a membrane. All right. And similarly, sometimes you see smaller objects diffusing around, and eventually, they join together, and this would be an association event. So these are, as far as I'm concerned, the kind of first examples of where we can actually follow dissociation events and association events in the context of a bilayer. Now, admittedly, these are not transmembrane proteins, these are membrane-associated proteins, but it at least shows that in principle, these kind of measurements are possible. All right, so I'm getting to the end. I hope I've convinced you that shining light on proteins is a worthwhile thing to do. You can measure mass, you get information about structure, you can do really simple things like question or investigate the purity of your sample, you can quantify interactions, and ultimately you can really start to use this to study dynamics and mechanisms. And from my perspective, I think we're really only at the beginning, right? I find this quite remarkable. 2007, this is what a cryo-EM structure would look like, and now you can see hydrogens, right? And the reason why that happened over 15 years, because people made better detectors and better electron sources and incredible advances in data analysis and got better and better at sample preparation. And, in my mind, the same kind of thing would be required here, maybe not on that same scale, but in principle, if you build more suited detectors and got the optics right and improve the data analysis and the sample prep, then you could go from a situation where we were, maybe a year ago. This is where we were a year ago. This is where we are today in terms of experimental capabilities. And I think this is where we could end up. And I've done this, I've gone back to the proteasome, right? And so here you have all the different constructs and then interactions and ubiquitins and all this other stuff where you would expect it. And of course, once you get to these kind of resolutions, now you can start investigating complex mixtures. And then you can start following processes like this. Because of course, all of these things have different masses, right? What do I have? We can identify mass. How much do I have? You count the molecules. How does it change? How do your distribution change as a function of time? What interacts with what? You can distinguish different complexes by mass. All right, so with that, I'm done. Huge thanks to my group, because they're the people who actually do the work and are collaborators without whom we'd just be studying BSA. People gave me money, and if I may, I'll say we've got a bunch of positions available, and we're looking for people to come and join the team. So if you're interested, email me or go to the website to find out more. And with that, I'm done.
Fantastic. I will applaud on behalf of everyone here, because obviously we're virtual. So I hope nobody's offended that I'm usurping their position. Absolutely fantastic talk, as I'm sure everyone agrees. The floor is open for questions. If you'd like to leave a message in the chat, that's perfectly fine. If you would like to raise your hand and ask a question in person, that's also okay. Please feel free to jump in at any point. I'll start off with one fairly obvious one. Is there any prospect for making this work on a substrate that isn't glass? So, you're comparatively limited to a planar surface that kind of it sits at your sorry.
I think you're back. No, you're gone. You were briefly back, now you're gone again.
How about now?
Yes, we have you now.
Okay, yeah. Sorry about that. My headphones just turn off randomly. I have no idea why.
Yeah.
But yeah, so you've got your planar surface, and you're doing your subtraction and your careful light monitoring. Is there any prospect for, say, interrogating the surface of a cell or something like that?
I mean, you can do it. I mean, you can do it now, right? You put it on, but the problem is you're just going to get gigantic signals, right? Because, I mean, to give you an idea, so the glass roughness that you see, we think comes from about 10 nanometer diameter holes in the surface of the glass that are about 100 nanometers apart, okay? And those little holes produce a signal that is about 100 times bigger than the signal of a single protein. So if those kind of holes produce that kind of signal, you can imagine the kind of signal that you would get from a cell. Yeah. It's just astronomical. Now, again, that in principle doesn't have to be a fundamental limitation if it doesn't change, because you can do the subtraction. The problem is that it does change, because it only needs to change a tiny little bit, and then in this differential imaging, you're toast. You can no longer extract the signal that you're after. So to make a long story short, if you have a homogeneous interface, this will work. As soon as you have some kind of heterogeneous structure that is changing, you're really going to struggle.
So, I mean, that kind of gets to the essence of the question I was driving at, I think. Is there a prospect for, say, incorporating, 2 ultrafast pulses spaced femtoseconds apart to kind of freeze frame the motion, or is that just an infeasible amount of correction to have to apply?
No, I think it's a good idea. The problem that I see is, you would have to make sure that you actually get scattered photons that you detect from individual pulses somehow. I haven't thought about it that way. I mean, my fear is, so we use quite a lot of light, something like 300-400 milliwatts. And once you get to short pulses, then of course you're not only confining the energy spatially, you're also confining the energy temporally. And I think at some point you're going to start smoking things. But I have to think about the that idea. In principle, that could work. I'm not entirely sure yet how that rhymes with our background subtraction approach. But maybe then you don't need the background subtraction approach. So worth thinking about. But I'm afraid I don't have the capacity to immediately switch that on and figure out how it would work. But in principle, you're absolutely right. If you can synchronize it somehow, you might be able to reduce the background.
Cool. Okay. I'm just going to check and see if anyone's got their hand up. No, please feel free. I noticed several of my group members in the chat. You know my feelings on insightful questions at talks, so stick your hands up, people. And we have a question from Glenn. Glenn, would you like to ask a question, person, or I'll give you 5 seconds to turn your camera on if you do, or mention it. 5, 4, 3, 2, 1. Oh, he is typing. Hang on. Let's see if I promote Glenn, then he can turn his make a presenter, change. Oh, action failed. Lovely. Thanks, team. Oh no, there we go.
Okay, so I can see the message at least. And the question is, are there any benefits to contrast from applying additional high-pass filtering of the image in software along with the filtering and the optical path? Yes, is a simple answer. And I actually personally think that the biggest advances in technology over the next 3 or 4 years will probably be brought about better data analysis. Because if you think about it, so we're detecting blobs on top of a shot noisy background. So that really reminds me of cryo-EM, right? Blobs on top of a shot noisy background. And what we do at the moment is we measure each particle independently, right? And then get the signal, and that signal goes into a histogram, and then the histogram gives us an answer. So imagine you did cryo-EM and you did that, and you get absolutely nothing, right? It would be completely useless. The reason why you get structures out is because there's some phenomenal algorithms out there that do classification, right? And through the classification, you can eke out tiny, tiny, tiny differences between subpopulations if you have lots of events. Well, we certainly have lots of events, right? But we haven't quite gotten to the stage where we can eke out these subpopulations. So I think, you know, a lot of these resolution improvements that I talked about may well be enabled at some point by actually, you know, some kind of classification of the data rather than, you know, looking at individual particles. So that's one aspect of it. The other one is there's lots of additional noise that isn't shot noise. And this kind of high pass, low pass filtering is something that we already do in order to try and get rid of fringes and all kinds of other stuff that is messing with the signal. But even there's lots of opportunity for improving the data analysis going forward.
Cool. We have another question from Lucia. Lucia Petro, would you like to ask a question in person, or? I'm seeing no typing, so I shall take that as a no. So Lucia's question is, thank you very much for the great talk. I wanted to ask if this can be used to detect the folding and unfolding of proteins. So presumably mass stays the same, but conformation changes.
I don't know yet. So the effects of shape that we've seen to date are very weak. So for instance, you take an antibody that's 140 kilodalton that has a very specific shape. And then you take a globular protein at 140 kilodalton, and they will give you essentially the same measurement. We're still kind of trying to figure out what kind of changes you get when you completely unfold something, or you know, if you have completely disordered proteins. It's just we've struggled so far to really get really good samples and sufficient samples so that we can generate those statistics. Because between any pair of proteins, you're always going to have variations based on our measurement accuracy anyway. And so the question, you know, in order to answer the question, we're going to need statistics, a little bit like we had lots of proteins, sequence mass versus, so we're going to have to do, you know, different degrees of disorder, different conditions, and get there. There was a theoretical paper out recently that suggested that there is a change in polarizability with structure, but that it's very small, which kind of fits what we observed. So I think one day, yes, but we may not quite have the measurement precision yet in order to be able to tell the difference.
Cool. Just check for hands up again, because I don't want to miss anyone out, obviously. Yep, no hands up. Oh, Lukia's typing, so she says thank you. I was intrigued by your 300, 400 milliwatts at the sample that you mentioned earlier. Oh, sorry, if Lukia, I will promote you to a presenter if you did want to comment on your previous answer. They're a presenter now, by the way.
Oh, thank you very much. I just wanted to thank you for your answer.
Okay.
Pleasure.
Well, thank you for your question. So going back to your 300, 400 milliwatts, obviously that's quite a lot of power. That's not an infeasible amount of power, but that's the kind of irradiance that you start to see absorption from biomolecules. So you can generate fluorescence with 1/10 of that amount. You can start to physically cause events to happen in the biomolecule. Is that a concern, or are we working in a regime where we're saying, the effect is going to be so minuscule from this absorption, bond breaking, restructuring, that it's basically going to be lost in noise?
It's a good question. So, I mean, we have instruments that operate at different wavelengths. Some in the red and some quite near the UV. We see very little differences in the distributions that we measure with these instruments. They're very similar. So if the blue light was blowing stuff up, I would expect to see some kind of these changes. The other thing is that, what would have to happen, I think, is that just the light going through the sample would have to have some kind of general impact on the sample, because, our detector acts a little bit like a sticky patch for flies, right? Or a glass. It's just stochastic, whatever just diffuses and lands. Okay, and as far as we can tell, what we see on the surface is just a representative of what you have in solution. Yeah. So what you would have to do somehow is, during our integration time, you would have to blow stuff off or blow it up before it even binds. And I don't, because everything is non-resonant, right? We're still a good way away. I don't think we're actually depositing enough energy in these proteins in order to make them fall apart. Or not, at least not on the time scale on which we measure.
Sure. But
sure, if you put actin on and you turn the laser on and wait for an hour, it'll say chow chow eventually. Yeah. Right? It's not going to be happy.
Cool. Okay. Yes. One last one that I wrote down as I was listening along. What is there a kind of optimum reflectivity of the coverslip surface? Like, is there anything to be gained by, say, having an anti-reflection coating on there, or making it out of diamond so the reflectivity is really high, or what's the physics behind that?
I mean, if you think about it in a way, our little mask is just it's a non-coverslip way of making the coverslip less reflective. Right? That's essentially what we've done. And so coating coverslips would certainly be an option to get less background light and therefore increase contrast. The reason why we haven't done it is because you have to coat the coverslips, which also shouldn't be such a problem. But at the moment, we just pull them out of a box and we run a lot of experiments. And once you start doing all the coating, it just gets expensive. But that's not to say that that isn't a good approach. So it's certainly something that you could do, and I think that would be beneficial. At the same time, the reflectivity of the interface is about 0.5%. So to get it significantly lower, I think is not necessarily trivial with a coating. Also because we're illuminating at a range of angles, right? We're coming through an objective and the light gets, maybe not focused at a high NA, but still get focused. So your AR coating would have to work equally well for a lot of angles, which also might not be trivial.
Cool. Yeah. Cool. Okay. Well, I think we've reached the end of our slot. I didn't have any more in my questions in my list. So if anyone has any last-minute questions before we close the session, just checking. Nobody typing. No. In which case, then it remains for me to thank Professor Kukura once again. Excellent talk, as I'm sure we predicted at the start, and we were definitely sure at the end. Round of applause on behalf of everyone else. So,
thank you.
And the meeting or the session is